Hi, everybody. I'd like to thank Dr. Foster for inviting me to share my work with you today. I'm going to share with you some of the work that we do in my group in Halifax, Nova Scotia at Dalhousie University. And today I'm going to talk to you about how phytoplankton allocate their protein resources in response to environmental change. And so, as you'll see today, the way that phytoplankton allocate their protein resources can help us understand their ecological roles and their biogeochemical roles in the ocean. So I'm lucky to get to conduct this work with a great group of young researchers in my lab over here, as well as a great bunch of collaborators that have helped us with various aspects of the work that I'll present to you today. And most of the work that I'm going to talk about, all the work that I'm going to talk to you about today was done in the Southern Ocean. So our collaborators for these projects are based in the US as well as throughout Europe and parts of Canada as well. So I'm grateful to all these researchers for moving these projects forward with us, and I'm happy to share some of our results with you today. So the reason why my group is interested in protein in particular is because protein is the major biomolecule in phytoplankton cells. So in this little cartoon here, you can see roughly the breakdown of different biomolecules by mass in a cell, and you can see that protein is the vast majority. So what this means is that protein constitutes most of the cellular resources in a cell. We also know that protein is really important for understanding how plankton cells interact with the environment because they can help acquire nutrients, for instance. They also catalyze really critical chemistry that helps phytoplankton do what they do in the ocean. So by understanding protein expression and allocation patterns, we can get a real sense for how these phytoplankton are succeeding in their environment and how they respond to change. So specifically, we're interested in looking at how the allocation of protein resources might change under different conditions. So how might protein allocation to photosynthesis, for instance, or ribosomes or resource acquisition change as a function of changes in environmental conditions? So if we can figure this out, this will help us understand the biochemical strategies that help plankton succeed in the ocean, give us some insight into their ecological strategies as well. And really what I'm most excited about is that it gives us this mechanistic understanding of how phytoplankton use resources. So in order to articulate this a little bit better for you all, I'm going to take you down to the Southern Ocean, in particular the coastal Ross Sea and McMurdo Sound, which is shown here in this black dot. So if you go south from New Zealand, you'll end up here. So what we did was we went to the sea ice edge in this beautiful spot here, and we conducted weekly protein sampling for 4 weeks from approximately the same location. And we also did a bunch of work to characterize the chemical environment in which these plankton are growing. So one of the things that we did was to conduct these bottle incubation bioassay experiments where we isolated plankton in these bottles from the surface water, and we amended them with various different micronutrients like vitamin B12, which is required by phytoplankton and produced by bacteria, as well as metal micronutrients, iron, and also manganese. So we added these in combination and by themselves, and we observed how this changed the response of the phytoplankton. So I'm showing you here the response in production of chlorophyll, which is a pigment that can serve as a proxy for phytoplankton growth and physiological status. So what we can see here is that when we added these micronutrients early on in our time series here, we did not see a change in chlorophyll. So really very little change in the phytoplankton community through manipulating these micronutrients. But then as we progressed through the season, what we could see was that these micronutrients started to become important later in the growing season. So we saw that when we added them in this 3rd week here, we increased the production of chlorophyll dramatically with the addition of each of these 3 micronutrients. So what this suggested to us is that the plankton were replete for these micronutrients at the beginning of our time series, and then they became stressed for lack of multiple micronutrients at the same time, so B12, manganese, and iron. Those were all impacting phytoplankton and causing them some stress towards the end of this growing season here. So our question is, how does phytoplankton protein allocation respond to this transition? And what strategies are these phytoplankton using to succeed, even though they're growing into this period of stress here? So the way that my group asks these questions is to use something called mass spectrometry-based metaproteomics. So what we do is we take our field samples, we extract protein from them, we digest those proteins into peptides, and then we can separate those peptides into less complex fractions using a technique called liquid chromatography that separates the peptides based on their similar chemical properties. We then interface those peptides into a mass spectrometer. And by measuring their mass to charge ratios, this mass spectrometer can help us identify peptides. So what it does is it first measures the mass to charge ratio of these peptides, then it adds some energy to the system, and that energy breaks apart the peptides into predictable pieces. So since the way that these peptides is predictable based on their sequence, we can, using a variety of informatic approaches, back out what the original peptide sequence was based on this mass spectrometry data. So mass spectrometers are getting better all the time, and even more dramatically, our ability to extract meaningful information from these mass spectrometry data is improving. So we're getting better and better at interpreting metaproteomic data in the ocean now, and that puts us in a position to learn some pretty important things, I think, about how plankton succeed. So the two approaches that we use in my group, we always use these in combination, is to examine both untargeted and targeted, to use both targeted and untargeted methods. So these untargeted methods allow us to ask the question, what proteins are here and how does their relative expression change? Whereas these targeted approaches allow us to ask questions about really specific proteins and peptides in particular, lets us ask how much in absolute quantities of a specific peptide is in a sample. And to do this, we need to know the identity of that peptide before we get started. So my group uses these approaches simultaneously and usually together. They help inform each other. And what I'll do now is show you what kinds of insights we can get from this time series here. As we know, we're transitioning from a micronutrient replete to a micronutrient stressed condition over time. First, we'll take a look at the community composition that we're able to reveal using mass spectrometry-based proteomics. So what I'm showing you here is a taxon-specific peptide abundance. So what we did here to calculate this was to pool all of the peptides that were assigned to dinoflagellates to assess their abundance and sum them up, and that's what gives us this this data here, or these data here, where we can see that the abundance of dinoflagellates is pretty high in this system and is even potentially increasing a bit by week 4. We can also see that diatoms, for instance, they're increasingly important as time goes on, as we get into this micronutrient limited system. And we've got a group here called haptophytes, and this is mostly Phaeocystis. It's important at the beginning of our time series and becomes less important through time here. And we've got various groups of bacterioplankton that we might be interested in as well. So what we can do now with this is ask what protein allocation strategies are helping these diatoms succeed here in the face of this increasing micronutrient stress that they're facing. So we'll focus on diatoms for a little bit. So in order to ask this question, we might want to know, are diatoms increasing the amount of resources that they allocate to manage B12 stress over time? So the way that we can ask this is by first figuring out what we expect diatoms to do with their protein allocation when they're facing B12 stress. And so we can turn to the literature, and to some experiments that I conducted back when I was a PhD student in Max Saito's lab. And we can see that under high B12 conditions, diatoms express low concentrations of these two proteins here, CVA1 and MET-E. MET-E is a protein that replaces the major B12 requiring enzyme in diatom cells. So by expressing more MET-E under low B12 availability, they're responding to B12 stress and trying to replace some of that B12 requiring enzyme with an enzyme that does not require B12. And in the case of CBA1, you can see it's low expression under high B12, high expression under low B12. You can see that this protein is also increasing in the face of B12 stress. And we think that this protein is involved in B12 acquisition. So what this represents is increased protein effort for B12 uptake. So we can then ask, do we see these same signatures in the field? And that's what I'm showing you here. So we've got our time course from the beginning of the time series to the end here. And I'm showing you here the concentration of MET-E protein relative to the abundance of a particular protein from diatoms that we think doesn't change very much. So this gives us a measure of how highly MET-E is expressed in diatoms. And that's shown here in these gray squares. And we're also looking at the amount of CBA1, so the B12 acquisition protein, as a function of total diatom protein here. So what you can see is that both of these proteins increase over time. So we are in fact seeing that diatoms increase their resource allocation to manage B12 stress over time. So we can ask the same question about iron. And so we know that this protein shown in black here, ISIP2A or phytotransferrin, is really critical for managing iron stress. It's involved in iron uptake. And what you can see is that the expression of this protein increases over time. And so this means that diatoms are devoting more resources to managing not only that B12 stress, but also iron stress as we get later in the growing season. So the question that I had after observing these trends was, what might be some trade-offs? So are there proteins that decrease in expression in order to allocate these additional resources to coping with that micronutrient stress? And so that's exactly what we see here. We see fewer resources are being allocated to photosynthesis amongst diatoms as we go from early in the growing season to late in the growing season. So this suggests that there are fewer resources remaining after they've changed their protein allocation to cope with that micronutrient stress. So what we're seeing is that in order to manage the transition into micronutrient stress, diatoms are devoting fewer resources to photosynthesis and increasing the resources they devote to manage micronutrient stress. So if we take a look again at what our protein abundance patterns are telling us about the success of these different groups, we can see that diatoms are increasingly important as we go forward in time in this time series here. So this strategy that the diatoms are using, this trade-off between coping with micronutrient stress and decreasing the abundance of photosystem proteins, it appears to be successful. So this gives us a bit of a window into the strategies that these diatoms are using to succeed in this later time point here when we know they're experiencing micronutrient stress. So this led me to think about what are some other strategies that plankton can use to succeed in this environment. And so what I want to do now is sort of compare the response that these diatoms have to the response of a very different type of plankton group, the SAR11 group here, which are important bacterioplankton. And the SAR11 are important throughout the world's oceans, I think, and they're present in often low but sustained concentrations throughout the ocean. And so what we can see here is that that SAR11 contributes relatively the same proportion of protein throughout our time series here, suggesting that it's a consistently important part of the community, but that its abundance isn't changing too much. So what I want to do now is compare the response of diatoms to the response of SAR11 in terms of which portion of their proteome is changing as in response to changing environment here across our time series. So that's what I'm showing you here. We've assigned each peptide from these two groups coefficient of variation. So this is asking how much does each peptide change over this time course? And we're showing the peptides that change, if peptides have a low coefficient of variation, they don't change much. If they have a high coefficient of variation, they do change a lot. And what we can see here is that there are more proteins and diatoms that are changing a lot relative to SAR11, which do not have many proteins that are changing. Much more of their proteins are staying consistent throughout this time course here. And over here in the next panel, I'm showing you the fraction or the amount of peptides, essentially, that are environment independent. And so what we mean by that is that they're not changing over this time course in the eukaryotic taxa and the prokaryotic taxa. So amongst the eukaryotes, the diatoms have the lowest fraction of their protein that does not change in response to environmental conditions here. So diatoms have the most dynamic proteomes. And we can see that SAR11 here amongst the prokaryotic taxa has the highest fraction of peptides that are environment independent, so or not changing as a function of this, as a function of time in this time course, where we know that there are some important environmental changes happening, namely to the micronutrient environment that they're experiencing. And so what we can see here is that there are different strategies for success here. So we've shown that the diatoms have a dynamic and responsive proteome, and that's consistent with what we understand about their ecological niche as well. And then in contrast, we've shown that SAR11 has a particularly static proteome, also consistent with what we know about its lifestyle and its strategy for surviving throughout the world's oceans. So these protein expression patterns and their allocation strategies, the way that they change these protein allocation strategies through time can give us insight into the mechanisms that these different groups use to succeed in the environment. So I want to just take us quickly back to the diatoms for a minute and tell you about an exciting new way that we've been looking at our metaproteomic data. And so what we've been doing is combining these metaproteomic assessments of protein allocation in diatoms across this time series with this model that my PhD student was able to create that optimally allocates diatom protein resources to different functions based on environmental conditions like micronutrients and nutrient concentrations, and optimally allocates these protein resources to generate the highest growth rate possible. So what we can do now is combine our measurements of the way that diatoms allocate proteins, plus the environmental conditions that we're able to measure, to start now to predict what the growth rates and the elemental stoichiometries of the diatoms in our system are going to be using this allocation model. And that's what I'm showing you here. So what we can see are the inferred growth rates using the model and the input of this geochemical data, as well as our protein allocation measurements from this system. We can see that there's higher growth rates early in the season and lower growth rates later in the season. And that to accomplish those growth rates, we're seeing higher iron quotas at the beginning of the season than at the end of the season. So I think we're just at the beginning of understanding what kinds of insights these models can help us generate about what's controlling plankton growth and how to predict the impact of plankton growth in a changing environment. But I'm really excited about this sort of new way to use metaproteomic data to help us understand what's going on in the ocean and to help us understand the strategies that plankton are using to accomplish what they do in the ocean. So I'll just quickly summarize. I think I've shown you that plankton groups use different protein allocation strategies to respond to the same environmental conditions, and that these allocation strategies are broadly consistent with what we know about different ecological strategies that these plankton use. We've shown that we can use metaproteomics to give us a great window into these different allocation strategies. And now we're starting to use allocation models with this metaproteomic data to help us understand growth rates and elemental quotas of plankton in the ocean. And so with that, I'll just again thank my group, and in particular Scott McCain here, who contributed a lot of the measurements and the models for the work that I've talked about today, and Eldon Rowland here, who contributed the rest of the measurements, and the Allen Lab in particular for access to that McMurdo Sound field site and to some metatranscriptome sequencing that enabled us to do the data processing and arrive at all these protein allocation strategies. So thank you all for your attention, and I'm looking forward to answering your questions next week.